Hello people, welcome back to Molecular Biology and Biotechnology with Lucy. Today we'll be talking about DNA profiling techniques, also called DNA fingerprinting techniques, which is the process where we extract DNA and analyze for reasons such as forensic studies, paternity testing, population genetics, and so many other applications. So we'll be looking at the four main methods that we have. That is RFLP, STR, mitochondrial DNA, and YSTR. So we start with restriction fragment length polymorphism, which we commonly call RFLPs. So usually this process will use restriction enzymes, also called molecular markers, to cut DNA at specific recognition sites. So restriction markers have their origin from bacteria, which is a defense mechanism for bacteria not to be infected by any microorganism or organism that contains DNA. So it's able to digest the DNA of the invading pathogen. So we usually now have this replicated in this technique where we utilize restriction enzymes that are going to cut our DNA at specific recognition sites, which are our restriction sites. So the enzyme that we commonly use in forensic analysis, for example, is HAE3, and it cuts DNA at GGCC sequence 5' to 3'. So it will recognize everywhere that there is this sequence and it will cleave. Usually this method will require a large amount of DNA and it does not work with degraded DNA. So that's a limitation of RFLP. It requires that you have sufficient or large amounts of DNA and the DNA should be double-stranded. So now usually because everyone's DNA is different, like the sequences of GGCC will be different along the length of your genome, the enzymes will cut different places and thus result into different length fragments. And that is what we call length polymorphisms that are restriction fragment. And usually the DNA will cut with restriction enzymes of different lengths because this is the reason there could be deletions of DNA between restriction sites, such that for one individual, there is a deletion at maybe the GGCC at a certain particular point. So then it means it won't cut at that point. There could be an insertion that will increase the length of the fragment. There could be a mutation that makes it not be recognized. And there could be a variable number of tandem repeats. The repetitions of this could be very different, and that will also result to different lengths. So essentially here I have put an example of what we mean by cutting using restriction enzymes. So if, for example, we have this fragment of DNA1 and this fragment for DNA2, in this case, we are using EcoR1 as our restriction enzyme, and it recognizes the region of GAA, TTC, GAA-TTC, GAA-TTC, and the same on the opposite side or on the complementary DNA strand. So what will happen is that once it recognizes this region, it is going to cleave. So there will be that cleavage site, this one and this one for DNA1. And once you cut here and here and here, it means you yield a 4 kilobase pair fragment and a 5 kilobase pair fragment. Now in DNA2, if there is a mutation here, you can see that here instead of GAA is GAC. It means that it cannot recognize this region. And it means it can only cut at the edge here and also at the other edge there. And so now the fragment will be longer. It will be 9 kb. So when you run a gel, you'll find that you're going to have now for DNA 2, it's just a 9 kb fragment. For DNA 1, we have 5 kb and 4 kb fragments. Of course, this one saves molecules by their molecular weight or size, and that's why this one will travel slowest, slower, and this will be the fastest because it's the lightest. So RFLPs, once you develop a gel, we call that DNA fingerprints. Okay, and this is another example where we have 5 samples that have been cut using a certain restriction enzyme. And for number 1, it has yielded 2 fragments, and you can see the 2, the first one and the second one. For 2, it just had 1 fragment. That one, for 3, 2 fragments. For 4, 2 again. For 4 is 3 fragments, and for 5 is 1 fragment. And usually on the edges of our gel, we put what you call a ladder that has known size, so that you're able to also estimate the sizes of our DNA molecules. So that is what we call a DNA fingerprint that has resulted from RFLP polymorphism. So what is the process of, RFLP? First, you have obtained your sample. That is the very first thing, that you obtain your blood sample. After you have obtained your blood sample, then you're going to isolate the nucleated cells. This is DNA extraction process. Then you purify your DNA. There are various methods of purification of DNA. I'm going to place the video of purification of DNA or DNA extraction methods in this video at this point, so that you can also refer. After that, we are using restriction enzymes to get our different fragments here. So here we digest our DNA using now your restriction enzyme of interest. Once you have digested, you're going to now run a gel to separate or to sort out your different fragments into bands. Okay, so we now run a gel here so that we are able to have our size separated fragments. And then at this point, you also add what we call sodium hydroxide. We mentioned that in this process, we require DNA from this stage here to be double-stranded. Okay, so here we add sodium hydroxide to make our DNA single-stranded DNA. And this is going to facilitate the process of hybridization. Then after you have made it into single-stranded, then you transfer. Now what we call southern blotting, you now transfer. So southern blotting is that process that you combine electrophoresis, separated DNA fragments, you transfer them to a filter paper, and then you have detection by probe hybridization. So you place a filter paper such as nitrocellulose paper onto your gel. Then using capillary or electroblotting, you're able to transfer your DNA onto the paper. At this point, they are invisible and therefore you cannot see anything. Now, after you have done that, then you do what we call blocking. You block your membrane using bovine serum albumin or casein. Now, this is because not every place on your paper has DNA. In short, when you separate it, there are regions that do not have the fragment. So you are blocking those regions to ensure that when you put your probe, it does not unspecifically bind to those areas, because it will give us false positive results. Then after you have done that, now you have your hybridization process where you add complementary labeled probes that are going to target the specific regions of your fragments. Once they have hybridized and they are labeled either with a radioisotope material or maybe digoxygenin or fluorescein, these are fluorescent materials, then you can now perform a visualization process. If you used radioisotopes, autoradiography. If you used a fluorescent dye, you use fluoroscopy. And now this yields something like this. Of course, remember the sides have the ladder that helps us in quality control, also in measuring of size. So you're able now to have this, and you can even put this on a computer for analysis to give you now an electrophelogram like that. But even at this point, you can still be able to do interpretation of your DNA fingerprint. So that's the process of RFLP. Now, something that is important, we've been talking about specific complementary probes. How do we determine these probes? It is because we have already done a mapping or a loci of RFLP markers. We have been able to locate chromosomal markers of RFLP where we can have digestion using restriction enzymes. So we already know the sequences of those regions. And here, usually on the human chromosomes, we have a marker at chromosome 1, this one, D1S7, another one at chromosome 2, another one at chromosome 4, another one at chromosome 10, and another one at chromosome 17. So usually, the loci are located on chromosomes, and therefore each locus, of course, has two alleles, the one that comes from the mother and the one that comes from the father. So you can see we usually have 5 commonly used probes in forensic, that is in forensic applications. And you can just use one locus or many, depending on how your analysis is. So a single locus is used and are selected because they occur at only that particular place, and they are hypervariable between persons, like no two persons will share how the DNA fragmentation is going to occur once you use restriction enzymes. And that means it can highly differentiate one person from another. It's only highly identical when you're having identical twins. So I have an example here of how you can interpret an autoradiograph. So we have a victim of death here, and we have his blood here. And then we have a defendant who we have gotten as our suspect. So there is that blood that we obtained from the defendant's clothes, and we were also able to obtain blood directly from the defendant after they were put in custody. So from here, it is actually possible. I don't know if you are able to check and tell me if this defendant is actually responsible for the death of the victim. Is he responsible or not? From what you can see, I can give you a minute or two. Is this defendant responsible for the death of this victim? From this autoradiograph, we have ran gels. This is the victim's blood. Then this is blood on defendant's clothes. There was one that was from the shirt, a region that had around 4 micrograms, and another region that had more blood, around 8 micrograms. So we have ran the gel of that. We also had blood gotten from his jeans, which was a little bit not very good quality. And then we had blood that has also been obtained directly from the defendant. So I am sure now you have an answer. So the answer for this particular question is yes, this defendant is responsible for the death. Yes, and the reason why yes is because if you look at the victim's blood here, you can see the bands that have formed. Now the clothes that are coming from this defendant's clothes are actually matching, from the jeans to the shirt, are matching the victim's blood. However, the defendant's blood itself, which you obtained, does not match the victim's blood. So it only means that the blood that was found on the defendant's clothes came from the victim. And because the victim is dead, then there is a high chance that this defendant is the one who is responsible for the death of the victim, because the blood of the victim is on his clothing. So that is one of the ways in which you can interpret autoradiographs of RFLP. Now, the probability of two people having exactly the same DNA profile is between 1 in 5 million to 1 in 100 billion. I mean, this is rare. to have people sharing the same DNA profile when you use RFLP is very rare unless you have two, like now an identical twin. That is usually the case. Now you're likely to have chances that they are sharing their DNA. Here's another exercise. You can do it during your free time. You can just pause the video as you are going through, and then you can tell us, in this case, which child, if any, can be excluded as being the biological offspring of this father here.